Hello everyone, I hope you're all doing well. My name is Ronald Carrasquillo and today I'm here representing the broader Bristol Squibb team that worked on the development of a semi-continuous drug substance process where we heavily leverage PAT to understand and address the obstacles encountered during the project's progression. Before I go into the specifics of the work, I'd like to really provide some context as to the background of my group, and I hope that will set the stage for where this talk will go. I'm a member of the Reaction Science and Engineering group, and that's within chemical process development. And in there, we aim to combine first principles reaction understanding and engineering fundamentals to deliver robust processes for API production. We use a number of different tools, but today I will be discussing the use of PAT and flow reactors in a stage appropriate manner. By that, really, I mean that we heavily probe the process up front. We invest very heavily using a large number of orthogonal techniques. Largely, these are real-time techniques in order to define control strategies around the process and essentially be able to define the quality attributes that really should be set up around the process and finally trim those back down as we move later onto development so that we can rely on instruments that are broadly available and very robust, and whose readout can really be correlated directly to the techniques that we used over at the beginning. With that in mind, the process that we tackled was the production of diazo compounds. Specifically in this case, it was EDA. And so this reagent was really to be used as an intermediate for one of the steps in our route. EDA, as a member of the diazo family, is really well known for its decomposition, which is exothermic. It's an explosive compound and it has a really low onset temperature for thermal runaway. We actually went ahead and measured all of these features and properties of the compound, but ultimately, really what that's really points to is significant safety concerns as we move from the lab into some sort of large-scale production where we aim to use this compound. So because of this, we thought, well, maybe running a flow process really is very worth our investment and our attention since it has the potential to really address some of these issues. And so we were able to use widely a very well-known synthesis scheme for this, and essentially we adopted it to be performed in flow. And so essentially what we did here was we use aqueous solutions of sodium nitrite and glycine ethyl ester, and we used toluene to be able to extract the EDA into that organic phase as soon as it is made. The rich organic then that is coming out of this reaction would go into the separation. And that is performed using Zaiput separators. And I'll go into more detail of those in a moment. We essentially save up the organic effluent out of the separation and wash it with a base wash, which gets aged for some period of time. And we perform a second separation using another Zaiput membrane separator. We again retain the organic and we pass that over to an IR unit to essentially be able to assess the concentration. At the end of it all, really aim to use this reagent, the EDA, to perform a cyclopropanation for a starting material and produce obviously the product that we intend to. So each of those units operations obviously goes along with the real world tools that really enable them. And essentially these are all highlighted here in this slide. And so we have tubing, pumps, membrane separators, as I mentioned, chip mixers that are relevant for this scale, and FTIR flow cells. And when you put them all together, you end with something that looks a little bit messier, but still translates quite well from our scheme that we showed before. And so essentially this really ties up very well into that scheme that I just mentioned. So I did mention before that I would go into membrane separators, and I think it's important to go over how this work. In essence, this technology is really just attempting to exploit the interfacial tension between the phases, and the aim is really to allow only one phase to permeate through the membrane while the other gets retained, and that's how you achieve your separation. Because we can measure and calculate the interfacial tension of various mixtures, we can really benchmark the efficiency of the separation across the flow range of the unit. And in our case, particularly, for this process, we were essentially operating in this region highlighted over here. And that for us means that there's very little effect on the flow rate into the actual breakthrough or retention that we have of the organic and aqueous. And so to us, this means that we're in a really good operating space for our process. Now, in order for us to assess the real-time performance of the flow process, that is the in-process concentration as well as the establishment of steady state, we use an inline IR probe. The method that we used monitored an isolated EDA band as well as a toluene band over here, and these are ratioed to assess the concentration, and we were able to calibrate this response just using QNMR measurements as shown here by the red dots. Having this information was really key to understanding if the process was in control, as well as when to proceed to charge the EDA into the cyclopropanation pod, as we really needed to understand, if the quality and attributes of our stream were met. We were quite fortunate in that we were by no means limited to just IR, and we actually observed matching trends using IR, Raman, and even UPLC as I'm showing here. So the trends for all these are essentially just identical. And you can really, in this process, pick and choose which one you want to go with. But for the sake of simplicity, I'll be largely focusing on IR results. Now that we had sort of established sort of a good set of analytical tools to monitor the process and the production of EDA, we then turn to obviously applying the continuous process into the downstream cyclopropanation. So at the onset of our work, it became very clear that the EDA that we were producing was not performing as well as the commercial counterparts. That to us really was seen in a decrease in the conversion that we were able to observe of the process, and ultimately we were able to address this using either kickers of EDA, but the majority of the time we even needed kickers of both the catalyst and EDA. To us, this really showed that really the issue here was the catalyst was being poisoned, and of course one simple answer is to increase the catalyst loading, but as the catalyst and the ligand were the largest cost drivers for this step, we really went out on the hunt for the root cause of the difference in performance. So to that end, we analyzed the BMS EDA against commercial lots, and we use a variety of techniques. We really found no differences until we actually dug very deeply into the IR traces. So specifically, the IR for the BMS EDA shows in purple here a band at around 2340 wave number, and this is not present in the commercial lots that we purchased. And so when we then now go ahead and apply some other techniques to this, then to the BMS EDA, we were able to use mass spec to show that the samples that we were producing in-house had elevated signals for both NO and NO2. And so, as you think about the signals, it is possible that they arise really from the decomposition of nitrous acid, and this is a byproduct of the EDA process that we're using here. And there is in the literature clear evidence that this species can partition quite well into organic solutions. And so this sort of ties everything together as to why then we're seeing these effects here. And to us, this sort of was a good finding in the sense that at least we have a lead, and also more importantly, that in this case we know that these impurities, these NOx impurities, are likely a dissolved gas. And so with that in mind, then we can go ahead and try some things that are simple but maybe quite effective at addressing this. And so we went ahead and took a stream of freshly prepared EDA, and we split that in two, and essentially degassed one of them and did nothing to the other. And as you can observe here, essentially when you do not degas, you have a significant portion of the starting material left over. Whereas when you actually do degas for a few minutes the stream, then essentially you're able to achieve full conversion in here. And so this really points to this a very dramatic change in reactivity, and again, sort of an indirect confirmation of the effect of removing this species, this NOx that is in solution. So now that we knew that we could address the problem, I think we had to go back and think as to how to implement this in flow. Luckily, this sort of problem is very much in the realm of chemical engineering, and so we tried two approaches. I'm really not going to dwell too much on the degasser itself because we found it actually didn't work for us. But I will be discussing in detail the work that we performed on the stripping column. And the reason for this is that stripping columns are really nothing else than pad columns, but maybe they're not widely known to people. And so essentially they're composed of obviously the column itself, but more importantly the inert packing material. And the simple idea or the general idea is that you will feed a contaminated liquid through the top, clean feed of gas, in this case that would be nitrogen for us, through the bottom. And essentially they will contact each other, aided through the surface area that is imparted into the liquid as essentially it spreads through the packing. And what you're meant to get at the end of it all is essentially a then dirtier stream of gas that has picked up some of the impurities and a cleaned stream of liquid that is then suitable for your purposes. And so ultimately, this is really just a countercurrent operation, and so this is not uncommon, and it's applied quite a lot in a lot of different scenarios. However, coming back to then the process scheme, since we know we can detect this impurity by IR, it really makes a lot of sense to pipe in this stripping column right before the IR detector so that when we start to degas our stream, we're able to assess the amount of it that we've actually removed. And so that we can maybe try to do this in the lab without experiencing poisoning or any stalling of the reaction. But and of course, if you do this, like, we were able to actually implement this and saw very positive results to where we achieved this. But thinking forward, what we really need to understand is how do we understand the controlling features that govern the behavior in this unit operation right here in the stripping column. And so, for processes involving gas-liquid exchange, this typically means looking at the KLA, the overall mass transfer coefficient, which is really just a rate of mass transfer. In this case, there's, I would say, a few parameters that I'm showing here, and I'll add then even a few more equations. But really, what it all boils down to for a fixed piece of equipment like we're using is that essentially if you modulate your gas flow rate, because your liquid flow rate and inherent constant are all system specific, then you're able to actually achieve the trend that we're seeing here, where you're gradually able to remove more and more of the impurity as you operate at higher gas flow rates. And so what's really nice is that then we can translate all of this through the equations that I'm showing here into an actual KLA value. So again, the mass transfer coefficient that sort of tells us the rate of mass transfer from the liquid into the gas, and so a rate of removal of this impurity. And so for us, this really was important to calculate because we wanted to really be aware of the realities of scale-up. And so really that means that we can't guarantee that a CMO will have a stripping column, and they may need to run this as a batch operation. And so we went ahead and tried to show how you can do this. So in flow, essentially on the left here, what you have is that essentially, since you're operating at some condition, you either have initially some of the material that is poisoned and full of the impurity, or you don't have it since essentially you're operating at steady state, and you can very clearly see that delineation. And in a batch process, obviously time is changing as you operate it and it moves forward. So essentially you start out with a good deal of the impurity, and over time that just decays as you remove more and more of it. The thing that is important to be aware of here is that however you decide to operate this, the same rules and the same features are really governing this process. That is, for example, in a batch process, you're really depending on the surface area to volume ratio of the bubbles that essentially are dispersed in the liquid to be able to clean up and remove that material. Uh, Whereas then in a flow process, in a stripping column, you're really taking advantage of the high surface area that are provided by these packings that are very custom and very highly engineered to then give you a much higher transfer rate for your process. And so that's really what I guess I want to leave you with on that front. There's really no difference in how you operate except for that. So in the same manner that I showed before, as we think to sort of scale up, things then obviously have to change a little, but the skin to the process is pretty similar. So we have the reaction with an equipment that then is a little bit larger, but it's a tube-in-tube reactor, so it's essentially just jacketed to allow for heat exchange. Then we have Zaiput membrane separators. It's actually look essentially the same as you scale up. So there's not much interest in there to show. We have static mixers, which just allow us to get pretty good mix in there, since we do have a biphasic stream. And then the streaming column I already highlighted before, even though I will show it pretty soon to you once we put it all together. And then again, we have IRs and Ramans, and so because it is a larger flow setup, we decided to pipe in both of them. And finally, we'll be tying this into a 5-liter reactor where we essentially be performing the cyclopropanation. So, as is typical with increased complexity that happens as you get from scale up, things look a little bit messier again, but we have essentially the things that we can sort of make sense of. So essentially, we start off with our 3 pumps, with our 3 streams that get fed into a manifold, going then into a static mixer. That static mixer, we have actually temperature probes at predefined points to be able to track the process. That stream that is already mixed then gets fed into the jacketed coil. Uh, that coil then essentially goes into the separators. We then perform the second wash going into a second separator, and we even can incorporate some mass flow meters in between them that are being read elsewhere. The effluent of the second wash then goes into the stripping column when then it gets degassed. And then finally, we essentially use some pumps to move that fluid around from the column, send it to the IR and the Raman, and then right into the cyclopropanation pod. So at this point now, with the amount of information that we have, we can start tying back into the claims I made initially. Specifically, we use temperature probes placed at different lengths of the reactor. That's sort of I'm highlighting here. Uh, and the idea was to really compare this against the IR trends that we've had, a lot of good information come out of. Uh, the temperature trends, particularly if we focus on this magenta one here at the top, are really remarkably similar to the IR trend. So essentially, we are able to at some point establish steady state, and it does oscillate around some mean value, but it is quite steady. It is very responsive to changes that are intentional in the system. Let's say we shut off one of our key reagents. You can very quickly detect that through a drop in temperature. And this really goes to highlight that while, we very much invested in tools that are quite expensive and not necessarily available everywhere, we can dial this back down to something as simple as a probe that just measures temperature. And in this regard, a temperature probe is quite an effective PAT tool for this type of process. As to the performance then of this reaction at a 100-gram scale, whereas it concerns the cyclopropanation, we were able to actually do this several times. actually 5 times in the range of 100 to 180 grams. And we observed throughout addition-controlled kinetics. Essentially, the product gets consumed, generated as you add EDA into the pot. And you only really start to get accumulation of EDA at the end. And so bear in mind, these are normalized intensities. And so while this looks like a pretty high peak, it's actually quite small amount that we're really accumulating at this period. Uh, Interestingly here, across all our runs, we didn't see any catalyst poisoning, even at the original levels of catalyst amounts. And to us, this really just meant that uh, this was a confirmation that our efforts really produced a robust process that we felt we could probably move forward to larger scale. So with that in mind, then we moved it forward and we performed 3 batches at 10 kilograms each at a partner CMO. I'll briefly go over the setup there and just mention that the scale of equipment was planned around using modules. And so you have modules for the pumping, modules for the reaction, modules for the separation. And so these were essentially just interlocked together, obviously incorporating much more stringent controls and safety interlocks. But overall, the process was, I would say, unremarkable in that it behaved just as we saw at the lab scale, and this is extremely good in my opinion. The vendor did decide to perform the degassing operation in batch. However, we were able to predict to within the error of our measurements, the time that it would take to actually achieve that degassing. And so this sort of went to show how really like it is important to collect this information and be able to twist it and manipulate it as you can and see fit to fit the occasion that you actually meet in the future. So now to sort of then wrap up here and finish my talk. I really want to go over a quick recap of what I showed you today, which is that we used PAT and we dug very deeply into the process. And the aim really was to understand when steady state was established, as well as to determine the CQAs that needed to be met around the reaction and the separations. Because of our early heavy investment and the amount of data that we collected, we could really be able to tie it back into very simple tools that were actually quite powerful, such as temperature profiling and mass flow meters. And ultimately, this really culminated in a successful translation of the process that went from milligrams to grams and then to kilogram scale. And so with that, then, I will end here at what I think is the most important slide, where the names of all the people who were instrumental to this work coming to fruition are present. And I want to thank you for your time and for your attention. And I really hope you enjoyed this talk. And wherever you are, I hope you have a good rest of your day. Thank you very much.